Hey everyone, it's Ryan Flynn. I'm here with Devan Shah, an old colleague of mine and friend. We worked together at Resilience a couple of years ago, and now we're both doing our separate things. I'm doing a lot of communications and journalism work in healthcare. Devan, do you want to give a quick overview of your company and what you're working on?
Yeah, sure. Yeah, I'm the founder and CEO of a company called Renovate Bio here in the Boston, Cambridge area. We're about 3 years old, focused on mRNA engineering and design, and with sort of a B2B business model. And yeah, we work with a range of biotechs and pharmaceutical companies to help basically make their RNA programs more effective.
So Devin and I were chatting this year about a variety of stories that came up, both positive and almost some controversial negative about RNA medicine. We thought it'd be great to do a quick overview for people just about update of what's going on this year. There's so many different stories. Devin, let's kick things off. What is the top three things that are, in your mind of the big stories involving how RNA medicines are either, changing healthcare, potentially could change healthcare, or the industry itself?
Yeah, no, I think it's a great question. This year, there's obviously been a lot of crazy news ongoing, and a lot of volatility. But the top three, I would say, there was this story a few months ago about a baby named KJ who was basically potentially cured of his genetic disease, but certainly seems to have really initially promising efficacy data. Um, uh, from a basically N of one customized mRNA based gene editing, base editing approach. I believe this is kind of like the first time this has ever been done in terms of a truly customized for one patient, rapidly developed program. And so that was really exciting and promising. We can definitely get into that. And then
Yeah.
more recently, there's been some M&A deals, which I think the market's really investors, operators have really been waiting for, just given a lot of depressed valuations out there. But pretty exciting science and also pretty prominent companies in big pharma stepping up and buying, RNA companies and both gene editing with Lilly and Verve, and then, in CAR T and in vivo CAR T with this company called Capstan out of San Diego and Abbvie. Yeah, happy to kind of run through those, but those are some of the more promising ones, that are kind of showing me signs of a market rebound. That's, slowly but surely starting to happen, but there's certainly other nuggets out there that are also looking pretty promising on the data front.
Great. Well, let's start into this baby KJ story. It was fascinating. I remember seeing this when it came out. This is where they use CRISPR gene editing to essentially correct a deficiency that this child had. And tell us a little bit about, the science behind it. How do they actually do this, and what is different in this approach versus other attempts to say, correct gene editing in infants or babies?
Yeah, I mean, quite honestly, gene editing, so far has been done more in, adolescence, just because of safety concerns with the FDA. And it's still a fairly early field overall, although there's, one approved product that Casgevy, which, folks may know about, and that involves taking the cells outside of the body, the hematopoietic stem cells, editing them, knocking out a gene, and then putting them back in, basically, so that in this case for sickle cell. And that was basically developed by CRISPR Therapeutics and commercialized with Vertex, and that's now out there on the market. The challenge with that is it's very difficult to manufacture. It's very expensive. It also involves conditioning, which means you're basically essentially getting chemo to destroy your existing blood stem cells. So the promise of a lot of these newer approaches with that often are using mRNA is doing the gene editing entirely in the body. And so the mRNA is expressing a nuclease, and there's different types of nucleases, and they have different pros and cons. But this is, to my wit, the first time gene editing's been done in a baby in the U.S. in like an N of one sort of circumstance in a customized fashion like this. And I think it was because this disease is super rare, and then it's lethal. And then I think also the maturity of the technology in terms of what data is out there with adolescents was such that I think, and the groups who are working on this between Penn, Danaher, some folks at Berkeley, they have the credibility to kind of develop this in a very fast manner, but also in a way that I think they were able to convince the FDA the risk reward ratio was right. And to their credit, they did it in 3 doses. So they did it in one really low dose, and then they increasingly upped the dose, which
In the
would
same patient?
for the same patient, for baby KJ.
Mhm.
And so the reason they did that too is from like a conservative, sort of risk mitigating sort of strategy, because they didn't want to have, obviously anything bad happen to the baby. And they weren't sure because this is novel on multiple fronts, as I just mentioned. And so they started with an extremely low dose, and they didn't see any toxicity or elevated liver enzymes or anything that was concerning. And so they were able to raise the dose just to make sure that they can drive enough editing rates to help ameliorate the disease. And using this specific type of nuclease called a base editor that was invented in David Liu's lab, and is a really powerful platform technology, which has a couple or actually multiple companies kind of built around it. So in any case, yeah, that was sort of the circumstances, and the 7 months end to end, I think, goes to show the kind of the speed of manufacturability and development for these precision tools across gene editing and mRNA. And then, I think also at the FDA cell and gene therapy roundtable they had a few months ago, it was pretty clear, the FDA commissioner, RFK, and, Jay Bhattacharya, they were all pretty positive and excited about this as a way forward, for genetic medicines for rare disease, genetic disease patients. So hopefully this, can help accelerate the field more broadly.
Yeah, you mentioned one thing I want to just dive into a little bit. You mentioned that they stepped up dosing for this baby, and traditionally with regular or traditional gene therapies, it's a one and done approach. You can't redose because the body is now immune to that viral vector. So how is this able to work? And is this sort of the promise of CRISPR and RNA medicines where you can deliver multiple doses or step up doses if the first one doesn't work?
Yeah, that's a great point. I'm glad you brought that up. So that's exactly right. With the viruses, typically there can be these sort of antibodies created against what's called the capsid, the protein shell. With the mRNA approach, the mRNA is encapsulated in what's called a lipid nanoparticle. So there's like a mixture of lipids, and these can cause some inflammation, but so far it doesn't seem like they are causing a basically like an antibody response specific to the lipids that prevents redosability. Intellia actually, as a company, has proven this out too with more patients, like maybe a dozen or so, for like a second redosing. But you're totally right. The dream and like sort of the aim is one and done. But sometimes, well, in this case, because it was a baby, they wanted to start really conservative and then to drive the total volume or percentage of overall cells that you want edited to ameliorate a disease pathology. In this case, they made the decision to do it 3 times. But I would say, generally speaking, the goal will be one and done as much as possible to avoid any, risks. But
Yeah.
the thing is you can redose with this approach, at least seemingly so far. Granted, the overall end of this data package is still pretty small, so it's definitely an area that a lot of these drug developers are continuing to like watch carefully, whether that's the dose or whether that's how you optimize the lipid nanoparticle to be like less inflammatory. But yeah.
I mean, I think that's sort of the other thing too. People talk about gene therapies as being a one and done. However, we don't really have, lifelong data for a lot of different diseases because they are so newly approved.
Yep.
And this could be potentially a good breakthrough where, say you correct a condition, but then it comes back at some point, you can redose with this versus a gene therapy where you no longer can. So, and a lot of people that I've seen, I've been surprised to see that people haven't necessarily gravitated towards gene therapies for sickle cell or hemophilia. And some of the reasons that patients give is they feel like they have their condition under control somewhat with their existing medications, even if there are still a lot of bleeds for hemophilia and hospitalizations. The concern is that if they get a gene therapy and it doesn't work or it reverts at some point, that they're stuck. So it seems like this could potentially answer one of those patient conditions or questions, concerns about gene therapy, especially as we go from beyond a disease that could literally, end your life at birth versus something that's a livable condition, although not necessarily one that you might not want to live with.
Right, right. Yeah, definitely, definitely. I think it does provide some flexibility. And then there's people also working on what's called epigenetic editing. We don't have to get into all of the details on that, but a potential benefit of these other approaches with epigenetics might be you can turn on and off the gene in like a very durable fashion. But certainly, I think that's also, to your point, why the siRNA therapies have had a lot of success. And Alnylam, who's pretty close by me, is doing really well in certain genetic diseases because they last about six months or so per dose, and but you could stop if there was an issue. and it's not permanent. But in this case, the editing will be hopefully permanent. But like you said, if from a redosability perspective, or if you did want to stop and it wasn't fully kind of there and you wanted to go back and revert to another therapy, at least seemingly so far, there's optionality there a little bit with the RNA LNP approaches.
Speaking of LMPs, you mentioned Capstan as a company that you wanted to focus on, and I actually spoke to them earlier this year for an article I wrote, or it published, or is going to be published soon, with evidence-based oncology about this potentially coming wave of in vivo CAR Ts. So, in immunotherapy treatment, that typically you have to get cells taken out of your body, they get refined, it gets manufactured into a more powerful drug, sent back to the hospital, and then you have to be watched for a couple of weeks to see if you don't have an intense immune reaction. And because of all that process, it's limited who has been able to receive the CAR Ts. The in vivo CAR T just basically makes the medicine in your body. I know this is a high level, maybe potentially inaccurate way of describing it. I'll let you correct me, but if you could give a shot that then makes the immunotherapy in the body, it could allow it to be delivered in many different places. And Capstan's one of the companies that is working in that. So why don't you give me your perspective on Capstan, and then we can talk a little bit later about in vivo.
Yeah, absolutely. No, I mean, I think that it's really exciting. I've been very bullish on in vivo CAR T for a little while now. But essentially, Capstan, has was able to raise a fairly tremendous amount of money as a preclinical company, about $400 or $500 million, given its roots from UPenn and some of like the top tier academics there, Carl June, et cetera. They had basically only had one patient in the clinic treated, and this was at a healthy human volunteers sort of study initially, and for lupus, going towards lupus, so for autoimmunity. But yeah, the data, hasn't really fully been actually announced, I don't believe, but essentially, I think it was enough, evidence for Abbvie to come in, step in, and buy them for 2 billion upfront, or up to 2 billion upfront. So certainly a pretty, meaningful deal for essentially what is a preclinical biotech, especially in this market. So I think it goes to show the excitement around, in vivo CAR, and I think it's because pharmas as well as biotechs recognize this is not going to be a niche commercial application. These are larger markets, because the CAR can be used for a variety of B cell mediated autoimmune diseases, of which there's many, and it can also be used for cancers. Just the way, the ex vivo CARs are kind of right now mostly used, and so there's like kind of a dual pronged approach that gets you to hundreds of thousands, maybe millions of patients. And so from that perspective, I think it's a top line and bottom line mover, needle mover for even these large companies. And so yeah, I think it is just going to be kind of like the start of a little bit of an arms race in a gold rush. But they were definitely one of the most capitalized in this early field, and there's several others, Orna, Tessera, et cetera. And there's a bunch of others too, not even just in the US and in China. So I do think the data sets here are going to start moving forward fairly rapidly over the next year or two. And I should also just say, that approach, the Capstan approach, is also using mRNA and lipid nanoparticles. The major difference is the mRNA is, encoding a CAR instead of a gene editor. So it's transiently producing this synthetic receptor. And then the lipid nanoparticle has been targeted to T cells inside the body, to your point of what you were saying, to make it more elegant from a manufacturing perspective. And there was a lot of work in R&D done to make that targeting effective, because otherwise these lipid nanoparticles generally kind of accumulate in the liver if left to their own devices. So they did a lot of work around, basically binding or, conjugating antibody fragments.
So could it go to anywhere in the body? Could you direct it, or does it have to go to the liver still?
So this is a topic of immense research and R&D right now across, a broad array of companies and academic labs to try to get these to go to different tissues that you want. Let's say you want to get to the heart or get to the brain. I would say right now it's still fairly limited in terms of what has been de-risked. The liver is definitely by far and away the easiest, and it's still not easy, but it's the one that's most kind of default. Liver, spleen, and lung, when you do IV, that kind of general system, this reticular endothelial system, is kind of what gets circulated. So you'll see some percentage distribution between those tissues, which is why if you look at the other clinical trials, lung is also popping up quite a bit for mRNA LNPs. And there's been some activity in spleen as well, but there's just less diseases associated with the spleen. So yeah, the T cells and hematopoietic stem cells seem to be next up. But I am talking to some groups and companies that are working on the brain and are working on the kidney and stuff, and it's just earlier. But so there's a lot of like R&D around how do you get that lipid to really go there. But yeah, the liver is still kind of where the gravity kind of takes it.
Yeah, and the reason why I ask is because, there was recently a patient death in a gene therapy trial, and I've worked at companies when this has happened in the past as well. And it's not like there's a direct link necessarily between the medicine and the death, but what seems to happen is that, especially with uh, weight dependent dosing, so you have to give a patient a larger dose depending on the weight, and usually with children, this is, 2-year-old to
Yeah.
5-year-old, a big difference, and that volume can cause potential harm, or can lead to maybe an infection or some kind of reaction in the liver that again causes a cascade approach that could lead to a death at some point. So the ability to direct medicine directly to where you want to go, whether it's a muscle, the brain, or somewhere else, could potentially be a big difference for the next generation of medicines in this space.
Oh, for sure. There's no doubt about it. I mean, delivery is sort of, very obviously like the number one bottleneck for the field, for the field's growth, essentially, because there's only so many liver diseases. I will say, I mean, this actually might be a nice segue to kind of, talk a little bit about Lilly Verve,
Yeah.
because, the interesting thing there is while Verve is delivering the mRNAs that encode a base editor to the liver, um, they're essentially treating heart disease by basically treating cholesterol. So the liver, I mean, the liver is a protein factory in general in the human body. That's just kind of how it works. It's got a lot of amazing functions, but in that case, it would be extremely hard right now with today's tech to get that, lipid nanoparticle and mRNA directly to the heart. So Verve was being clever by basically attacking heart disease, but just via the liver, given the delivery bottleneck. And so there are kind of cool things like that too that are workarounds. And so, I think, yeah, completely agree with you. Like having the ability to get it to the right organ that you want to is definitely the holy grail and will definitely, help accelerate the field. But there are some workarounds for specific tissues that can act as depots, so to speak.
So let's dive into Verve. This is a great story. So they announced in April that they had positive data from their phase 1b clinical trial of Verve 102, which is an in vivo base editing medicine targeting PCSK9, a target that's been around for a long time. We have some biologics targeting that came out, I think, 10 to 15 years ago. I remember being a reporter at Bloomberg and covering that news. And what was sort of fascinating about the release that Verve put out in April was that they showed a mean reduction of bad cholesterol of 53% and a maximum reduction of almost 70%. And obviously, someone who I'm on personally on statins, but if you look at these drugs, having that large of a reduction is certainly an amazing achievement, and it's also a sustainable, supposedly long-term achievement. That it's not like you take a pill every day to reduce your cholesterol like I do with statins. It's a one and done shot that will continuously improve your cholesterol. And heart disease, I think, is the biggest killer in the U.S. right now. It has been for a long time. So if we could help with that with a single shot, you can imagine the excitement, and that's why obviously they were snapped up shortly after that news came out. And it's a really interesting story. So, why don't you give me your perspective on the medicine, how it works, and why it matters?
Yeah, and it kind of goes back. It's a lot of the same principles as what they did for baby KJ. And so it's also using base editing, and essentially inactivating the PCSK9 gene so that LDL uptake is inhibited. LDL kind of being like a real driver of in this case hypercholesterolemia, who are patients who are like super susceptible to basically cholesterol related pathologies and sort of the whole downstream cascade, which can lead to heart attacks and all these other complications. But yeah, to your point, the idea is this is one and done. There's a lot of people who could benefit from that. It's still early. That's just the initial clinical trial data, but I think the safety that's been seen, just not just with that, but also with what Intellia has done in some of the more rare liver diseases and ATTR and HAE, are showing that this should be likely scalable. And then I think Lilly saw Verve's, market cap as being appetizing where it was at. They traded down quite a bit just given the broader biotech economic struggles that,
Yeah.
we've been witnessing for the last year or two or three. And so, they came in and they already had a partnership with them, by the way. So they had a lot of insider info, I think, probably on where things were going. But, kudos to Verve too for executing and also pivoting because this trial was their second one, actually.
Oh,
They,
okay.
they made a modification to the lipid nanoparticle to get better uptake in the liver because their first one, it had some issues with that and it had some higher inflammation. So going back to what we were talking about earlier, so they actually stopped it before they saw that being an issue. But I think this also like goes to show, I think to my wit, this is like the first big pharma meaningful acquisition of like a gene editing technology in general or like of a new gene editing technology, like using CRISPR or a form of CRISPR. And so like all the other CRISPR companies right now are independent, like all the meaningful ones. And so I think it's the first time you're seeing big pharma say, okay, yeah, we see the commercial promise here, and that this could be a real, therapeutic modality that's a needle mover for us. So I think that's also going to help accelerate the field, which has honestly been just struggling a bit with all the challenges of clinical trials and FDA concerns around safety and also just around crowded indication spaces like you were mentioning where there's just lots of competition from generics or antibodies or siRNAs. So it's kind of I think a breakthrough on multiple levels. And one other thing too I'd comment is like, I mean, I think Lilly probably gets this really well, but with their GLP-1s and Novo probably as well. But you see all these extra benefits that happen from some of these like really, major, driver diseases of pathology. So, obviously obesity in the GLP-1 case. And then there are all these studies around how it helps with like addiction and like mental health, obviously, and all these other things, right? Kidney disease even. And I suspect with heart disease as well, we may see that. It's not entirely clear. I think obviously these things need to be validated in trials, but if you know people with heart disease, there's a lot of anxiety, right? There's a lot of like mental health pieces that come with that because it can always like blow up on you on any given day, and you just don't have a lot of predictability, right? And so I think there's potentially a lot of downstream positive repercussions too if things like this get commercialized, but they've still got a decent road ahead of them.
Yeah, I mean, thinking about expanding indications, there are even examples of people trying to use RNA for obesity, right? Reduction.
Yeah, there's a few different approaches out there. Wave I think has like an RNA editing program in preclinical development. They're moving forward for a target. And then on the siRNA front, I believe there's also some work going on there with like fatty liver. And then yeah, on the mRNA front, I haven't seen quite as much specific to obesity. I'm aware of like one or two preclinical programs, but yeah, I do. It's going to be hard to catch up with the massive success of the peptides and how high the bar they've set, is. I think we're seeing that a lot now with these next gen obesity kind of fast followers. But there's certainly a lot of things that people want to optimize for, like muscle retention or gaining muscle while losing weight, so you lose the right types of weight and you still stay strong, right? So I think
Yeah.
there's a lot of these things that are adjacent to it that can augment it as well. And I think RNA will undoubtedly play a role. It's just a question of like where's the fit? Where's the fit relative to the other types of therapeutics that are out there, and where does it stand out?
Yeah, I mean, especially as we're getting, this is a general, issue in overall in medicine, but if you have eventually an oral pill, GLP-1, that'll be a lot cheaper than an RNA injection that's a lifelong, it's a cost benefit analysis, right? One and done shot that could change your life. But if you look at the current crop of GLP-1s, the injectables, I mean, most people, I think 60% quit within a year just because a lot of them quit because of the GI side effects, which can be pretty intense, nausea and everything else. So, I know there's a lot of different approaches of people developing next generation therapies that eliminate or try to eliminate the side effects and also retain muscle. So that's an interesting space to see how that goes forward.
Yeah, yeah. It's like, it's a fairly recent phenomenon of like a really long-lasting therapeutic, like a gene therapy or cell therapy. There's still only a few, diseases where these are really commercially being adopted. And a lot of those were also with viral vectors or just cells, and those came with potentially curative, often curative upside, but also potential, serious, side effects as we're seeing with like, this is Sarepta data, unfortunately, but also
Yeah.
there's been some other things with CAR T and cytokine storms with ex vivo. So, hopefully the RNA profile can be, efficacious enough to be curative, but also very safe. And I think if it's very safe, that will really help it take off. And so I think the safety bar is really kind of one of the key questions.
And that's actually a really good point. Something else I wanted to kind of circle back
Mm.
to. We mentioned how CAR Ts might are currently being explored in autoimmune conditions, and there's a lot of excitement around that. However, it's a different calculation from a patient's point of view when you have cancer, and you have a CAR T that could save your life again with a pretty incredible medicine. We have an autoimmune condition that's not life-threatening. And you're a young person, do you want to get chemotherapy? And if you're, a woman, do you want to be infertile moving forward because of this? And is the condition of your autoimmune disease as intense as that cost? And I think that's a market we haven't seen. These drugs aren't out yet. They're still obviously, really early stage, but is that something that, an in vivo CAR T or an RNA medicine could avoid? I mean, would you have to have the same kind of prep work to get an in vivo CAR T or RNA drug that you do with the current crop? Or
Yeah, that's a good question. I think right now the general consensus is they're trying to avoid that. They're trying to avoid what's called the conditioning step, which is essentially chemo radiation and basically clearing your old cells, which are dysfunctional. Like, in the T cell context, clearing your old existing T cells, or in the bone marrow context, clearing that your existing hematopoietic stem cells to make room for the new ones that have been grown and are engineered. So that's the goal. I think it's still early enough to see, to say definitively we're not going to have to use that. But like Intellia is not doing any conditioning, and they're the furthest along in vivo, editing. And they're now doing a trial with like multiple hundred patients. They'll have an interim readout, I think, later this year, and then the full readout in like a year or two. But so that'll be really big. But the data so far is pretty darn pristine. I mean, it's like granted it's still in like the 10 to 20 range of patients, but or like a couple dozen, but the safety has been there and the efficacy is really strong. And they have that out to like one or, actually several years now in terms of ameliorating the major effects of like ATTR amyloidosis and then this disease HAE. So yeah, so far it's looking good without, having to have some of those processing steps that make it really difficult in terms of the trade-offs. So we'll see for in vivo CAR T. I think some companies are thinking about still having conditioning, and it's just early enough that I don't think anyone knows for sure yet.
Yeah. And just in more in general though, having the in vivo CAR T going back to cancer, right now there's I think 60% of people that are referred to CAR T don't get them. And it's for a variety of reasons, but one of the big ones is again because they're of the process is required and the the monitoring that you have to do afterwards. Most of these drugs are administered at large academic medical centers, and there are only a couple of areas across the country. Last year, I worked on a report from McKesson at the state of the cell and gene therapy industry. And one of the things we discussed in that report was the fact that there's these CAR T deserts, basically large swaths of the country where you can't get a CAR T because you have to fly across country. And it's not just the flight, it's that you have to stay there, or until recently, you
Yeah.
had to remain for up to a month with a caregiver. So, imagine you're a parent with young kids. And some people pull their kids out of school to live near the hospital for a month, or how can you have if you have two parents, you can't have them both leave little kids home alone for a month. Or if you're an older adult, I've also heard sad stories where people are saying, my kids are grown, they have their own families, no one can spend a month of me living next to a hospital. Now,
Right.
thankfully, the FDA has pulled back some of the regulations on these. They're called reams, and now they no longer have to spend a month at the hospital. It's I think monitored for 2 weeks, which is a little bit easier. But still, the issue is that most of cancers are treated. I think 80% are treated in the community setting. And so when you only can get a CAR T at a major center, that's not where people are getting treated. So if you could do an in vivo CAR T in the community, I know we're a couple of years away from that, but that would certainly change that equation and hopefully expand access to these medications.
100%. Yeah, no, I think that's exactly right. It makes it much more like, drug-like in a lot of ways, and a lot more scalable, both in terms of the manufacturability, and we could talk about the cost of goods and everything, but it's dramatically less than having to take your cells out and put them back in, or engineer them, expand them, put them back in. And then, yeah, like you said, like which medical centers can actually access this and administer it. So, yeah, it's definitely, moving in the direction of being sort of, and I think that's why you saw Abbvie come in on just one patient data. I mean, typically pharma companies want to wait until like at least one, meaningful like phase two is done for like an acquisition of that size or larger. And with, more statistical significance, more patients in the actual disease setting. But I think that's why they were able to be so aggressive there, is just because they realized like, yeah, this can be scalable to a lot of people. And the data, clearly must have been good enough in terms of targeting.
I think we've had a really good conversation so far, and maybe we could finish on one it's mostly been positive, exciting about what could potentially change. Is one that you and I have been talking about how it's kind of infuriating, but the negative backlash to RNA based on misinformation. So I wrote about this for Cure, which is a accelerator incubator in New York that's backed by Deerfield, and there are, I think, maybe a dozen states that have tried to ban RNA medicines, and it stems from this kind of anti-COVID, anti-gene therapy perspective in the community. And it's sad. When I was head of comms for a gene therapy company a couple of years ago, we were trying to position ourselves as a leader in the category of gene therapy. And so, as part of my work, I was analyzing social media posts on using the phrase gene therapy. And I mean, there was tens of thousands of posts, and literally 99% of them were negative things about, Bill Gates is putting microchips in your brain through gene therapy, and it just all these absurd things. But there's a growing movement of people, and sadly, like these are questions I would never bring up in a conversation among people in any kind of industry event 2 years ago. But even at Bio this year, I had to ask people, what do you think about these laws trying to ban and then criminalize? I think in Minnesota, there the law they drafted based on comments from a Florida hypnotist, of all things, wanted to put people in jail for 20 years for possessing or distributing mRNA medicine. So give me your perspective. I mean, you work in this field. This is something that obviously we're all excited about, but I mean, do
Yeah.
you come across these sentiments? What's your perspective on that?
Yeah, I mean, um, yeah, this is a tough one. I mean, because it just got the way it became, sort of in the public's, you know, you know, sort of, you know, you know, the way the public kind of perceived mRNA was all related to COVID, and all related to the pressures to get, the vaccines out. And so I think unfortunately got heavily politicized very quickly. And, meanwhile, the actual data and the efficacy was like extremely strong. Granted, there were some safety concerns and there's some things there that were not optimal, just how fast it happened the first time it being rolled out at large scale. But I think now, there was a major COVID crash after, in terms of the RNA market, in terms of, well, the virus mutating so many times, people just kind of being like, shrug, well, I'm going to get, COVID regardless at this point where I've already had it. It can't keep up with all the virus mutations and all the different, viruses that are out there now at this point, the COVID variants. And there's a lot of truth to that. But now the science has been advancing and it's becoming much more than just a vaccine story, like we just were talking about, right? Like mRNA can express any protein or set of proteins in the body. It's the fundamental medium between DNA and proteins. So that just gives it a huge flexibility, which was what I was always excited about with it. And it's easier than a lot of other types of medicines to manufacture. It's a much more like a small molecule in some ways in terms of manufacturing, compared to like viral vectors or cell therapies, or even antibodies, frankly. And so, yeah, that gives it a lot of advantages in how like the cost of goods can be, the scale up can be in terms of timelines, and unit economics. And then in terms of safety, yeah, I think the profile is getting better just as our science has gotten better in terms of our understanding of these, lipid nanoparticles, as well as the mRNA. I think it's the lipid nanoparticle that honestly was a little bit more of a black box.
Hmm.
And now we're starting to better understand some of the immune pathways that are being activated that cause inflammatory reactions that are undesirable. And so you're seeing people just be cautious about how they dose and do dose finding in these studies to make sure that they avoid that. But there's also a lot of optimization around these molecules to make sure you avoid that. But yeah, I do think like, it's always a range. Like any therapeutic or drug development, sort of, candidate is going to have a risk of a toxicity.
Yeah.
There's like very few that are just like completely, always safe, right? And so that being said, it's like, what's the benefit versus the risk? And increasingly, the RNA benefit risk profile is strong, not just in infectious diseases, but these other areas, rare diseases, cancer, autoimmunity, and that's a lot of people that can benefit and don't have reward and absolute risk reward that you have to kind of take into account. Ultimately, people can make their own decision. Doctors can make their own decisions. Maybe there'll be some places where it's not, looked well upon, but I do think, hopefully the overall data story convinces the vast majority of folks it's the rewards way outweigh the risks. And I
Yeah.
think it is.
Sorry, it
Yeah,
blinked out there for a second.
yeah.
But just to kind of close out on that positive note, I think you were talking about the fact that these potential bans are coming at a time when actually we're seeing a lot of incredible progress with what these medicines could do. There's potential for a pan flu vaccine, right? Every year, or cold and flu, these are developed based on what's the strongest strain happening across the world. 6 months later, we're hoping we get it right. They
Mhm.
usually don't work that well because by the time the flu hits the country, the strains have changed and a different one's more prominent. But if you could have, a pan cold or flu shot, that would be a huge benefit. I mean, a lot of people, that older people that do die, don't necessarily die from getting the flu, but it really does weaken your body, and you can have a heart attack or stroke a few months later that could be potentially tied back to the flu. So I know a lot of people don't like getting the shot, but if there is a pan one that could cover all the bases. And then secondly, you mentioned this as well, we're seeing some developments of RNA medicines for even cancer vaccines, right? This is amazing to have potentially a shot that could prevent you from getting cancer. So these are all exciting areas that I think are important to keep our eye out for.
Yeah. Actually, if I can comment real quick on the cancer vaccine, because that's interesting. And, I'm also thinking like, from a branding perspective, cancer immunotherapy is really kind of like a better way to kind of refer to it. Because frankly, it really is that. It's basically just arming your immune system with the better tools to be able to track and get rid of and eliminate, cancer before it becomes a major problem, before it becomes, unstoppable to some degree. But yeah, the data there is also looking increasingly promising. It's still, I think, a little early in TBD in a lot of contexts, but the Merck Moderna are putting a lot of money into multiple phase threes across multiple cancer types, lung cancer, melanoma, multiple others, and then BioNTech and Genentech with the pediatric cancer vaccine approach. Or cancer immunotherapy approach. So I think there's a lot of momentum there, and I'm actually starting to see with my own company there a decent pickup in interest in the R&D sector,
Oh,
as well
good.
as from high net worth individuals that may have loved ones or themselves suffering from cancer prognosis, and they're looking to take their health into their own hands and say, hey, can I fund something that can help me or my family? And so I think it's also potentially a path towards broader personalized medicine, and giving people more ownership of their health from like a molecular perspective, which is, I think, really cool. And the unit economics, I think, can make sense for that with RNA, more so than a lot of other modalities, which is really interesting. So yeah, I do think like, folks that are willing to read and understand the data are seeing like, yeah, this is the safety risks are much less than the potential benefits. And a lot of these, indications, unfortunately, you have a very miserable, sort of standard of care and just quality of life. So you really start to get desperate, and that's why we're trying to help.
Yeah, no, it's exciting, and it's a good point about branding or positioning. It's yeah, if you call something a vaccine, there's going to be a huge pushback, but if you call it immunotherapy, there'll be more interest. So I will say though, there was a company recently acquired this week, I think Vice, they do vaccines, and we're all kind of joking like vaccines in this economy, in this environment, like it's kind of the opposite of what you're expecting. But again, these are amazing medicines, and if we could do vaccines or immunotherapies or preventative immunotherapies for cancer, that certainly would be a big deal. Maybe kind of to kind of close it out, what are some of the things that you're looking forward to? Like, are there particular data readouts or things you have your eye on for the rest of the year?
Yeah, I think any interim data readouts from Intellia is going to be really, instructive for the field of in vivo gene editing because, as I mentioned, they're kind of leading that with their mRNA LNP platform that they've developed. Kind of the first company to really develop it for editing specifically. And it's a much larger trial that's been than what's been done. Two phase threes that they're doing. So I think they're going to have an interim readout, I believe, later this year. And then they'll have the full readout in a year or two. And they'll have to kind of continuously follow these patients to make sure it is truly a cure to the point of our earlier discussion where, is there any sort of recurrence of disease? But I think that's going to be very big. And then yeah, I think the the other areas are really just honestly a lot of early R&D making it into the clinic, just seeing companies because there's been such a funding supply glut, or just for preclinical companies in this market.
Mhm.
So seeing some of these companies that are more well capitalized bring their next gen mRNA programs into the clinic, for things like in vivo CAR T, in vivo epigenetic editing, next generation, cancer immunotherapies. I think that's also going to be important. The data readouts for those are probably going to be at least a year out, but
Mhm.
uh, but yeah, I do think the in vivo editing data should be very exciting, assuming nothing unforeseen happens on the safety side. But so far, if things trends hold, I think that could be a watershed moment.
Well, it was great talking to you, and we'll hopefully come back later this year with another update. There's so many things going on, so many data readouts and deals that I think it's great to kind of check in every few months or even every 6 months to get people in a sense of what's happening because it's happened so fast, it's hard to track it all. So this is really helpful, hopefully informative for a lot of people.
Yeah, likewise, Ryan. Always great chatting, and happy to do this again. And yeah, looking forward to it.
Thanks.
Cheers.